The current scenario in ART training across the world is of observation only, which although gives you a fair idea of how the procedure is being done, but does not give you the confidence of performing it independently.
I just finished a visit to Indira IVF Group in Udaipur, India, and I'm totally impressed by several things. But one of the things that I'm most impressed of is the way they just prepare themselves for the future and how they're expanding.
I'm here this afternoon visiting the Indira IVF Center in Udaipur. It has one of the most cutting-edge facilities that I have seen, including the perfect training for physicians and for embryologists. And I really think this is the future of how to train superb doctors and superb embryologists.
Embryology is confined not only to collection of gametes or making embryos, but it is about making euploid embryos to achieve a higher pregnancy rates. Embryology skills, lab culture conditions, gamete handling, and optimal environment are desired and necessary to achieve such results. The current scenario in ART training across the world is of observation only, which although gives you a fair idea of how the procedure is being done, but does not give you the confidence of performing it independently.
I just finished a visit to Indira IVF Group in Udaipur, India, and I'm totally impressed by several things. But one of the things that I'm most impressed of is the way they just prepare themselves for the future and how they're expanding.
I'm here this afternoon visiting the Indira IVF Center in Udaipur. It has one of the most cutting-edge facilities that I have seen, including the perfect training for physicians and for embryologists. And I really think this is the future of how to train superb doctors and superb embryologists.
Embryology is confined not only to collection of gametes or making embryos, but it is about making euploid embryos to achieve a higher pregnancy rates. Embryology skills, lab culture conditions, gamete handling, and optimal environment are desired and necessary to achieve such results.
So, welcome everyone for today's exciting session and with yet another speaker. So, I hope the video was visible to everybody. Just anybody can just unmute yourself and let us know that you can clearly hear our voice.
Yes, sir. It is clear, sir.
It is clear. Right. Thank you, Ranji. And yes, with me, Naval is there, and we really welcome Maria for taking out the time from your precious schedule. And yes, I understand, and I also thank SAR Healthlines for making this event happen today and for connecting you with us so that we can, have your online webinar session across Pan India. Yeah,
Great. Thank you, Ritesh, sir. And thank you, Dr. Maria and SAR, for making this session possible. Thank you, Dr. Maria, for removing time out of your busy schedule and being available for us today. Through IFA, that is Indira Fertility Academy, we are making consistent efforts to educate the masters in embryology, not only of Indira IVF, but all around India and anybody who wants to join around the globe to know and understand about the recent innovation, new topics, and some challenges in the embryology front, so that it is helpful and the academy, at the end, academy is to spread across the knowledge, which we are trying to do it in a very unbiased way to spread it across to everybody and make it freely available for everyone.
Yeah.
So thank you for being a part of our initiative.
So as far as the academy is concerned, just to introduce you a bit about, let you know a bit about IFA. So IFA is a subsidiary of Indira IVF, and it's an academic division of Indira IVF. So here we have training schedules not only for the embryologists, budding embryologists, but also for the clinicians who want to pursue their career in the field of reproductive medicine. Here we have short-term as well as long-term courses. The courses are designed in a way to have a very practical impact, so that each procedure we explain each procedure practically. We let through each embryologist to go through these procedures practically by on-hand training is given, imparted, and this is how we work in the IFA, in the Fertility Academy. So without wasting any time, so Naval, it's the time to introduce our today's speaker.
Right. So we have Dr. Maria Jose with us today. She is the head investigator at EV Valencia and FEV in Clivia. She is also a senior embryologist at EVRMA Valencia, which is one of the most advanced and scientifically thorough IVF unit. I have been myself on a visit in 2018 to EV, and I have seen the standards and the work which EV is into. And it's really a commendable task which EV is doing. At the same time, she is postgraduate teaching faculty at the University of Valencia and ADEIT. She participates as a principal. She has participated as a principal investigator in more than 10 competitive research projects funded through regional and national mechanisms. She has published more than 45 original manuscripts and book chapters, and her main topic of interest is in vitro rescue of immature GV oocytes, some part of which we will possibly hear today in our lecture, and the oocytes recovered from the stimulated cycles. She has also been part of cloning previously. She has her main topic of interest is also fertility preservation in cancer patients by ovarian cortex cryopreservation, and also the preconceptional genetic diagnosis of human gametes and gamete genome editing by CRISPR, the CS9 technology, which is in news and possibly would be the future. Like China has embraced it. We never know who all will be embracing it in the future to have the designer babies. But it's good to have you who have been an active researcher involved in so many such kind of research. And it will be great to hear you out today on the topic which you have selected. Thank you very much, Maria.
You're welcome. I get embracing with this. Thank you so much. Okay, thank you to all you for coming this afternoon. And I'm trying to explain you one of my pipelines of research about abnormal fertilization failures as a new approaches to optimize a cycle from a patient. Thank you to you all for inviting me, of course. And I would like to introduce you what is the content of my talk. I divide it into three main topics. The first one is to present you the fertilization process and how is the classical assessment of the fertilization. And then we dip into the abnormal fertilized oocytes. What are these kind of abnormal AFOs, called acronym AFO, in particular with the monoprionuclear, that means that contains only one prenucleus inside, or the triprenuclear ones that contains three prenuclei. in each one of these kinds of AFO, we are talking about what is its incidence, what is the possible origin, what is the developmental competence, and what is the genetic composition. To finally conclude a general conclusion on how to manage this kind of AFOs. So let's start with what is the fertilization. As you know, our target, our objective is to obtain a competent metaphase 2 oocytes. This is only the left picture, and it represents nearly 70% of the total oocytes retrieved. Morphologically, we can assess this mature oocytes because they contain this structure that you can see in this position. It's called the first polar body, and it doesn't take place in the.
Maria, sorry to interfere in between. I think you'll have to share the screen if you have the presentation, please. The slides are not visible.
Sorry. Really? Oh, I think that it was all the time. I'm so sorry. This is why I can. Sorry. This is why I can't see you. Okay. I'm sorry. Let's start again, okay? Can you see the screen or not? Yeah, I'm sorry.
Yes.
Okay, this is my lecture, but okay, and I divide the content in 3 parts. What is the fertilization, the process, and how we assess it. And we talk about the AFO, this is the abnormal fertilized oocytes, in particular with the monopronuclear oocytes that contains only 1 pronucleus, the tripronucleated one that contains 3 pronucleus, sorry. And inside of this, in every one of these types, we talk about what is the incidence, the possible origin, the developmental competence, and the genetic composition. And finally, I provide you, or we can make some final conclusions. Well, let's start with which is the fertilization and how we assess it. Well, after ovarian stimulation or in natural cycles when women ovulate, this is the gamete that we aim to obtain, the metaphase 2 oocyte. Morphologically is characterized by containing this structure. Can you see how I mark it in the picture? Yes. At this position at 11 o'clock, more or less. And this is the first polar body. It's a subproduct of the meiosis and does not participate in the reproductive process. And there is another structure, the oocyte itself, with no identified structure inside the cytoplasm. These oocytes represents around the 70% of the retrieved oocytes, and AIDS are target, right? So after that, we applied different fertilization techniques, either the sperm microinjection, the ICSI, or the gamete co-incubation, the IVF. So after this fertilization process, the oocytes were cultured for 16 to 20 hours in culture. In this picture, because of the time-lapse technology arrived to our clinics, we can learn about the embryo development. Here you can see the extrusion, the formation of the 2 pronuclei, one from the mother and the other for the father. The pronuclei contains the half number of our chromosomes, so 23 X for the mother and 23 X or Y for according to the fertilizing sperm endowment. So after this period of culture, 16 to 20 hours, zygotes or oocytes has to be assessed for both structures. The first one is the presence of the two polar bodies or the two pronucleae inside. This is the correct fertilization assessment as the Spanish registry and the Istanbul consensus applies, okay? And according to this, the benchmark used to be around 60%. This is the KPI ideal when you consider these two variables for assess the fertilization, okay? So in other association of clinical embryologists, as in UK, the fertilization is assessed by the presence of the two pronuclei, regardless the number of polar body. At this point, the fertilization rate that has to be achieved, the benchmark, has to be higher, maybe around 75, 80%. So in any case, the fertilization rate is reported an effective indicator about the gamete quality as well as operator competence, ranging from 60 to 90%. Well, but until 100, what happened with the rest of the oocytes? Well, around 15% of them were this not useful, right? They were not fertilized, as you can see in the left picture. They are metaphase 2 oocytes containing one single, the first polar body, and no structure identified inside the cytoplasm, or they were degenerated or atretic, or even they could be cleavage or activated. But there is another percentage that is the main, when I going to focus my talk, is this AFOs or abnormal fertilized oocytes, because contains different number of polar bodies and pronuclei than expected 2 and 2. So we are talking about the monopronuclear, the tripronucleated, that, okay, depending on the fertilization procedure that we use, it's the chance to produce 1, 3, or 2, 3 that I will speak later. Well, let's move to the monopronuclear or unipronuclear oocytes. Morphologically, as you can see in this picture, they are clearly identified by the presence of the first and the second polar body and a unique single pronuclear structure in the perivital, in the cytoplasm. Now I'm going to present you our work on rescue monopronucleated derived human blastocysts as a model to study also the chromosome topography and fingerprinting. So first of all, how does this monopronuclear come out? So the origin of these oocytes, of this AFO, could be haploid or diploid. It's another task different of euploidy and aneuploidy. Of course, a haploid is an aneuploid embryo, but a haploid is the correct gamete. So what is a haploid? A haploid means that it's an embryo that contains the half chromosome set that's expected as a species. So it's an embryo that is 23X or 23Y according to the origin. It could be parthenogenetic. It's called parthenogenetic when only is born from a virgin, that is the Greek meaning. It means that is when an oocyte starts embryo development, but without sperm participation. Genogenetic embryo concerns to those oocytes that start also with the embryo development triggered by a fertilizing sperm, but without genetic contribution. And on the opposite, we have another kind of haploid embryos that they are collectively called androgenotes. Androgenotes means that they are only formed by genetic content came from the father without any mother contribution other than the cytoplasms. Well, this is rare, right? It's very difficult to see that. it occurs. Another possible origin for these mononuclear embryos is the diploid. The diploidy will come from the non-extrusion of the second polar body. That means that instead of occur a perfect segregation of sister chromatids as a final result of the meiosis, these sister chromatids goes together and to the second polar body or stay in the cytoplasms and form one unique structure by diploid, only maternal by diploid by maternal side. Or could be a haploid, as I already say, but along development, it becomes, for a more stability, becomes into a diploid. Or even these monopronuclear zygotes could have their origin from fertilization artifacts. What does that mean? That means that the biology is a continuous process. So thanks to the time-lapse technology, we have assisted to the asynchrony in the prenuclear formation. So if you check the oocytes and a precise timing of your culture, for instance, at 28 hours, as you can see in the left panel of pictures, at 28 hours, you can see only one prenucleus. But if you rewind and you reproduce all the embryo development, you can see a two prenuclei 5 hours earlier. That means that there is some asynchrony. Around 4% of this asynchrony could be rescued, these oocytes could be rescued. And that, I mean, you can check the fertilization and 28 and you say, oh, I have one monoprenuclear zygote, but it's not true. It's because it was because an early prenuclear fading and asynchrony early prenuclear fading occurs in one prenucleus, but not in both altogether. So after reviewing the filmations, we can rescue these embryos as currently fertilized ones. The opposite is that you check the fertilization, but the prenucleus could appear later than the other one. So and at this point, to check the fertilization, the possibility to recheck the fertilization is a good point to reduce the number of monoprenuclear oocytes or even to increase the number of currently fertilized oocytes. That means those that contains the two prenuclei. And what is the incidence of these, the frequency of these monoprenuclear oocytes? So it depends on the fertilization technique that you use. For example, if you use the IVF, it's rare that obtain these monoprenuclear oocytes. The percentage is around 2%, but after ICSI, it's around 5%. Well, what about the competence of these embryos, of these monoprenuclear embryos? So we know that they are able to progress to the blastocyst stage. And even after time-lapse culture, we can assist to exactly same morphokinetics as all our sibling currently fertilized oocytes. So morphokinetically, we did not observe any significant differences between monoprenuclear or biprenuclear currently fertilized oocytes, and even on the rate to achieve this stage, the speed of reaching the blastocyst stage. And what is the potential? Can they give how long they could develop? So there are described many parthenogenetic cell lines derived from this blastocyst, this one prenuclear derived blastocyst. But and even live babies has been born even from uniprenuclear, monoprenuclear blastocyst derived from IVF or from ICSI. So at this point, we want to clear up in our hands, in our lab, what is the real blastocyst rate that we achieve after culturing these mononuclear and what is the genetic composition of these blastocysts, and even what is the parental origin of these oocytes. So in order to do this, we included a large sample of mononuclear oocytes. In fact, the number of mononuclear was 2,831 AFO, but after checking, we were only interested on those that contain the two polar bodies in the perivitelline space and one single pronucleus. That means that after all the population of mononuclear, we only rescue, or we were going to study only 40% of them. The majority of them contains only one polar body. And this is another population that we will discuss later. So after checking this 40% of two polar bodies and one pronucleus, we recheck the filmations, and around 4% of these monopronuclear were rescued because of this asynchrony on pronuclear appearance or pronuclear fading. So 4%, because of the time-lapse technology, we could rescue for patients 4% of this monopronuclear. So finally, almost nearly 200 of these monopronuclear zygotes were progressed to the blastocyst stage, which represents nearly 80% of culture mononuclear. After that, we studied this population of blastocysts in 3 experimental series that I'm gonna to present you. The first series contains, was formed by 26 blastocysts derived from monopronuclear, and we performed in all the embryo a FISH analysis, fluorescent in situ hybridization. And we study the chromosome 18, 13, 21, X and Y. And 2 different phenotypes could be assessed. The haploidy, as you can see, only 2 signals, only 1 signal for the X chromosome in that case, or for the chromosome 18. It could be also it could be diploid when 2 signals were detected, or triploid when 3 signals for each chromosome assessed evaluated. So these studies could inform us about how many chromosome sets has these blastocysts. So these ploidy studies were performed on blastocysts. The second series and third series consist on biopsy of these blastocysts into 3 pieces. The ECM by one side, and on the other side 2 different trophectoderm biopsies. Of course, there is no more blastocysts because the ending for these embryos is the study, and they were not transferred in any case. So from every single blastocyst, we obtained 3 portions of it: the ECM PC, the trophectoderm, and 2 pieces of trophectoderm. And what did we do was, in series 2, we study in the ECM and in the trophectoderm the NGS technique in order to assess the euploid or aneuploid condition of the blastocyst. And another part of the embryo was used to ploidy determinations. In the second series, we perform, as I has already said, we obtained 3 pieces of the blastocyst. One was assessed, the first one, for example, the trophectoderm was assessed for NGS, euploidy and aneuploidy. And the other one was assessed, was destined to assess the seroparentality composition. So is the embryo contains the 3 runs, that means that it could be AA, BB, or AB. It contains SNP arrays, single SNP polymorphisms from the mother, from the father, and from both, okay, from the both. Or biparental, that means triploid, when 4 runs you can see, or monoparental, when only AA or BB alleles could be achieved. So this is more or less the idea that we use. This is the blood from the father, the blood from the mother, this is the kid. So this is blood DNA from the father that you can obtain the genotype A or B. The mother could also be A or B, and this is the informative locus for this particular point. So when the embryo is AA, we put one that spot, BB another one, or AB 2. So this is the final panel from a heteroparental normal euploid embryo. This is the another when AA or only BB, and the bottom when AB, AAA, ABB, ABB, or BBB, or triple nucleated one, or triploid one, sorry. Well, what are the results that we achieved with this approach? The results were that after a study deployed on this large population, 35 blastocysts, we observed that nearly most of them, 50%, were mixoploids. That means that contain cells that were diploid, were haploid, mixing in the same embryo. The 45% were diploid, that is contains the 46XX or 46XY chromosome number, and only 8% of the blastocysts contains the haploid set of chromosomes. On the other hand, when we studied the NGS in the trophectoderm samples, we observed that the ratio was rather good as in the normal population, with nearly 60% of the embryos being euploid and 40% of them were aneuploid. And finally, and most interesting is that 40% of the blastocysts were not only euploid and diploid, but also contains genome from the mother and from the father. So it's a fingerprinting from the parentals, and it was interrogated into the embryo. Nearly another kind of embryos corresponds to those more interesting for me is that there were some uniparental blastocysts. So when only one of the parents is present in the embryo development is also possible. So what about these embryos that were genetically assessed? Are they able to progress in vitro? So as you see, there are many works, around 8 works, when these blastocysts that were analyzed or not were transferred, and 5 babies born after this transfer. In fact, last year, Clerk Asher at ESRE reported the largest size sample of monoprenuclear embryos that were analyzed according to IVF, obtained after IVF or after ICSI. And it's important for me because our population is close to that group in terms of the percentage of embryos that reach the blastocyst stage. We achieved 80.5%, and this group reported 70.4 70.4% of blastocyst rate. So we are on the same page. Slightly higher for IVF embryos, estimated at 34%. After analyzing these embryos, we are also our data are also in the same line as the 40% of the blastocysts analyzed, 37.9, were euploid and heteroparental, as we obtained, as I had already presented you, and 90% went uniparental as well. So after transfer, either blastocyst, euploid blastocyst, heteroparental coming from IVF or coming from ICSI, this group said that nearly 33% become pregnant and more than nearly 70 babies were born. In fact, one of them was Claire Jurses Nefer, as she told in her talk. Well, so what are my conclusions after this part of the talk about monoprenuclear AFO? So the morphological assessment of oocyte fertilization is helpful, but is not a definitive tool to determine the chromosome composition of the embryo. Blastocyst identification is also not definitive to assess ploidy, euploidy, and heteroparentality. And the most important thing is that 40% of these embryos are reproductively useful for the patient. They could be rescued and used for transfer. If you have not available the technique for heteroparental terminations, you can check those euploid blastocysts that were male, that contains the 46XY chromosome endowment, because they are most surely they are heteroparental in origin. But is not the same scenario for the euploid 46XX embryos that they are able even to implant, but they could came from parthenogenetic activation. Well, the next part of my talk corresponds to the tripronucleated embryos. This is now a new fashion in the reproductive to rescue also these kind of embryos, but I will alert you about the caution with this kind of AFOs. First of all, our group studied these two types of tripronucleated embryos according to the origin, according to the ICSI that used to contain one single polar body, the first one, and 3 pronuclei. Probably the extra pronucleus came from the mother. So that means that they are digenic in origin. 2 pronuclei came from the mother, and the third one came from the father. So this embryo is called two maternal or digenic embryos. Digenic. Another kind of tripronucleated embryos are those characterized by the presence of the first polar body and the second one in the perivitelline space. And of course, and it's assumed that that 2 of the pronuclei came from the father because of 2 sperm entry into the oocyte. And the cytoplasm of the oocyte cytoplasm has the ability to remodelate and to recondensate 2 or more sperm, as you, I'm sure that you have seen that kind of AFO in your lab. So these tripronucleated came from IVF, are diandric in origin, as we assume that 2 pronuclei came from 2 sperm and the third pronucleus came from the mother. Well, we culture these embryos, these tripronucleate AFO until the blastocyst stage, and the first point that we observe is that the blastocyst rate was significantly different between these two types of AFO. For example, for those derived from ICSI, the blastocyst rate was 62%, significantly higher than that achieved by those from ICSI, of those deandric in origin. But not even the blastocyst rate. Also, the morphology of this blastocyst is rather different. As you see in the upper picture, the blastocyst derived from tripronucleated ICSI-derived oocytes is a normal blastocyst that contains a perfectly defined inner cell mass and also a pretty good trophectoderm cells. But on the other side, on the other hand, blastocysts derived from IVF are totally another scenario. They has no inner cell mass and a really linear trophectoderm formed by very, very, really few number of cells. So when we analyze these blastocysts, these embryos, these tripronucleated embryos came from ICSI or from IFF, we observe differences, right? So half of the blastocysts came from ICSI were diploid and the other half were triploid. So contains or the normal 46XX or 46XY diploid chromosome condition. This is represented around in 50%, 50-50, and the other one has 69XXY or 69XXX, right? When we talk about the IVF, the proportion of diploid embryos is lower. They are the most frequent ploidy in these blastocysts is the triploid one. And when we assess in a trio format, that means we obtain DNA from the mother, DNA from the father, and a biopsy from the blastocyst, and we put all together and check some informative short tandem repeats, some genes, 10 informative genes from these patients. We check that effectively the embryo is heteroparental and composed by two from genes from the mother and from genes from the father. So as conclusion of this first trial, the tripronucleated derived blastocysts are quite different according to the parental composition in terms of morphology and ploidy composition. So tripronucleated ICSI-derived blastocysts, they can be self-corrected when they are cultured into the blastocysts, and we observed that 50% of them were diploid, but maybe they could be heteroparental in composition. However, when we are talking about, or we are assessed the tripronucleated blastocysts derived from IVF or deandric in origin, only 25% of them were diploid. So our interest on how to do was this IVF, these tripronucleated embryos that came from the gamete co-incubation. So it represents nearly 5 to 8% of the co-incubated gametes. And they are, as I have already said, they are limited in competence to develop to blastocysts and in diploidy. So we published it in 26, this paper, when we try to restore the bipronucleated stage in these abnormal A4 embryos. So we, I share with you this video when by micromanipulation, we positioned the tripronucleated and with a ICSI pipette insert into the oocyte, aspirate and grate and drag the prenucleus out of this zone, aspirate all the content, and after that, we assess that nearly 61% of the epronucleated zygotes survive, and after culturing them, we observe that 40% reached blastocyst stage, a percentage that was comparable to the similar to that observed in the control group, that means the currently fertilized oocytes, and significantly higher than that reported for those non-manipulated triple-nucleated eggs came from IVF. After analyzing diploid determination in a whole blastocyst, it was very amazing. It was, I'm so proud of this because it was a really nice picture that was selected for the front page of the Fertility and Sterility in that sample. And we assessed that this embryo were diploid. We restored the diploidy condition, and after analyzing 10 polymorphisms in a trio format, we also observed that this embryo were restored not only the diploidy, the euploidy, but also the heteroparentality. So what are these, how are these embryos competent enough to progress to a baby, to give a baby? So the answer is yes. There is a 1916 paper in which is referred the first live baby birth after the transfer of a triple-nuclear embryo by combination of some techniques for assessment. And even it also occurs in the nature. So as you can see in this picture, when two eggs are fertilized by two different sperms, you can obtain two twins, but they came from two different zygotes, so they are not related each other as well as brother or sisters, right? When one oocyte is fertilized by one single sperm and it divides and at the two cell stage, each blastomere, every one of the sister blastomeres develops independently of the other one, you can obtain sibling monozygotic twins, perfect twins. But there is another kind described in 2007 and in 2019 in Australia as well as in the USA, that it's called twins, ciszygotic twins. That means that when one oocyte is fertilized by two sperms and a tripolar spindle is formed. So after that, two twins could be born, but they are exactly the same from the mother endowment, the inheritance that came from the mother, as you can see here, print in blue, but they are different according to the father in orange or in green. So it occurs. So what to do? Test, okay? Well, so as a conclusion from these triple-nucleated IVF AFOs, I share with you that these derived blastocysts with high probability to be diandric in origin exhibit lower blastulation rates, only 20%, and a pathological feature with no inner cell mass, a linear trophectoderm. But after surgical correction of these zygotes, E-pronucleated zygotes, B-pronucleated zygotes could be obtained with a surgery efficiency estimated at 70%, and they are able to progress to the blastocyst at comparable rates with a pretty morphological appearance, and also after testing, it could be diploid and heteroparental. So my last part is around these triple-nucleated, but those that came from ICSI. As I have already said, it contains only one pronucleus and only one polar body, 3 pronucleus, 2 that came from the mother and 1 from the father. So what is our experience with these embryos? So as I have already said, they are able to progress to the blastocyst. Morphologically, they are a pretty lovely blastocyst, but only 20% of them were diploid, and most of them, more than 2/3 of them, were triploid. So this work was done when the time-lapse technology was available. So we want to retrospectively assess if there are any difference between diploid and triploid tripronucleated embryos. So the answer was yes, and this was a good news, because after culturing tripronucleated embryos derived from ICSI, we observed that triploid embryos progress more slowly than those diploid or correctly fertilized oocytes in all direct variables and also in indirect variables. So after analyzing 4 of these blastocysts that could be considered as diploid, we observed in a trio format also that all of them were heteroparental. So my conclusions according to this A4 group is that tripronucleated ICSI-derived blastocysts constitutes a heterogeneous population with only 20% of them being diploid, which could be identified by morphokinetics, and the blastocyst rate and morphology helps, but not is the unique or is not conclusive. And studies on heteroparentality and uniformity of the embryos are mandatory. So my final conclusion is that to know how many polar bodies and the number of pronuclei is mandatory to know how to handle with these A4 embryos and what are the possibilities or the possible endings of these embryos is required. To assess the asynchrony, the pronuclear asynchrony, and recheck fertilization is recommended. At this point, of course, the time-lapse technology plays an important role. And my opinion is do not discard A4. So it could be a reproductive useful. I know that this is a low percentage of the fertilized oocyte, but it is the 100% for some of the patients. So culture to the blastocyst is more than recommended, especially in monopronuclear embryos. Culture to the blastocyst, and euploid, aneuploid, PGTA, SA. Select those male embryos for transfer if your law allows. And when you are going to transfer female embryos, euploid female embryos, you need to assess that they were heteroparental in origin before transfer them. What about the ICSI? What about the tripronucleated embryos? So if they came from IVF, I recommend the surgical removal, culture, read the blastocyst, and perform a parental analysis. And if they came from ICSI, of course, culture, go to the blastocyst, assess the morphokinetics, and test for euploidy and parental analysis. And now there are another A4 coming up, figure out that those that contain 4 pronuclei, that recently Dr. Capalvo said that they have a pregnancy and a live baby born, and also those that not contains any pronucleus at the fertilization assessment. So the final message is that the key point is that now we are faced to a new concept that goes further than the morphological fertilization assessment, is the genetic fertilization assessment. So as my last point is to refer you to the next session that will take place next I think that in two weeks at the ESRE meeting, when now the A4 is coming up, is popping up, and there is an oral presentation which patients, there is patients that which is used to, in which kind of patients this AFO appear. So as you see in the main results and the rule of chance is that donorocytes exhibit a 1.32 times higher risk of producing mononuclear zygotes, similarly as those for vitrifiocytes, that also which improve their probability to obtain mononuclear zygotes. And when we are facing to oocyte and advanced age, the most frequent AFO that you can observe is the triple-nucleated zygote. So, and even this author said that is a risk increase by 10% for each additional year. So the AFO is now coming as an important part of the reproductive medicine. So thank you very much to you, to Indira IVF, for inviting me, for all of you for sharing this Friday afternoon with me, and especially thanks to Dr. Marcos Meseguer for propose me and give our science to share with all us. Thank you so much.
Thank you, Maria, for such a wonderful and enlightening session. In fact, as a trainer, I think that I had so much to gain out of this. As a trainer, I had always been telling my embryologists to discard the 1N and 3N embryos. But today you really changed my perspective of thinking about and of looking at the embryos, the 1N and 3N, as the culprit ones. And now I think it's a high time to shift to rethink before discarding such embryos, I think. Before proceeding, I would really like to ask Maria, how do you, first of all, you talked about self-correction. So when is the right time, how to decide whether to rely on self-correction mechanism of an embryo or to do some intervention like removing the one pronucleus? How do you decide about that?
Okay, it depends. In the early 2000, we used to, we use more the IVF than the ICSI. The time is changing, and we use more the ICSI than the IVF procedure. So when the triple-nuclear embryos came from IVF, they are always, or most of them, came from the spermic. And the sperm not only provides the genetic content, it also provides us a proximal centriolo, right? So to conform the spindle, mitotic spindle for the divisions, the oocyte has no centriolar endowment, has no centriolo. This is an evolutive mechanism to protect the oocytes to be parthenogenetic and not to need the males, So if there is some risk of women when there is a pathological aspect that now is going to be related with the teratomas. So if one oocyte inside the follicle starts to develop by itself without any male participation inside the ovary, it could become into all germinal lines, but in a destructured fashion, right? So it comes into a teratoma, ovarian teratoma, right? So the evolution makes that the oocyte has no centriole inheritance, okay? And who provides this centriole inheritance to the embryo? The male, the fertilizing sperm, right? So this is the normal, the regular, the natural, as things occurs, right? The sperm add to the embryo, goes to the oocyte and provides not only the genes, but also the centriole, the centriolar machine. Well, When two sperms enter, the tripolar spindle occurs. So the right way is to remove one of the paternal pronucleus. You're right? So yeah. It's the point. But when they came from the female, when they are dygenic, that means that the retention of the polar body occurs, we first study what happened without any intervention. So we were really amazing that according to the cleavages, according to the one thing that is called heterogonoic cleavages, that means that is asymmetric cleavages, the oocyte, the tripronucleated embryo that is dygenic in origin is able to refuse some irregular cells and only form a blastocyst to compact, even to blastulate, those that seems to be diploid. It remains to determine if these blastocysts are totally uniform or not. I'm not sure if I answered your question, because it's true. The first point is, okay, why do you not remove the surplus maternal one? But the first point was, what happened? Why we are going to check what is our population, what happened with these embryos, and we were really amazing with the self-diploidization. So we have not to do anything because they go self-diploidize. I'm not sure if I answered your question.
Yeah, definitely. And just a continuation of that, how do you, if at all you decide that, okay, we'll have to go for intervention or this invasive intervention, how do you decide which pronuclear to be extracted? How do we decide on?
This is a good point. Okay, first of all, with at random, you have the probability of doing okay, of doing fine, of doing all right, 2 out of 3. So 2 are male, are from the sperm, 2 sperm. So at random, you can remove to have a high probability to do it well. But the best point is because you have the 2 polar bodies, those pronuclei that they are geometrically nearest, more closer or more near the first and the second polar body, they could be with high probability maternal in origin. So you have to decide one of those that are far away the polar bodies. This is the point to select the blastocyst. Select the pronucleus to remove.
Okay. And by any means, is there any correlation between the size of the pronucleus and labeling them?
Good point. Yeah. Good point. It's true. There is some correlation. Some work said that there are two other morphological indicators to select the surplus male pronucleus. The first one is that the male is the largest one. They say that male is the largest one, but at the end of the syngamy, they are more or less all equal, right? So the point at what time do you decide to check? This is the point that you can lose that point or not. So if there is some asymmetry, okay, I remove the large one because I know that it's the father, okay? And sometimes if, for example, you have 3 pronuclei, but there is one that is not included in the group, so there is one that is isolated, there are 2 nuclei at syngamy, and one that is the lie that is not included in the center of the zygote. So they're not integrated in the trio is removed. If there is some largest is removed. if there are two, they are making the syngamy, and there is another one far away, the couple is removed. So there are some tricks that you can use for select the extra surplus paternal. But the last one is a good point. Yes, you are pretty good. Yes.
Right. So basically, the conclusion would be the polar body, the PN which is far away from polar body, remove. If they are distinctly separate, you can remove, and or if they are larger, then you can remove, right? But at the same time, after listening to this talk, I believe yes, there would be embryologists who would possibly try it out. Okay, that if they have 3PN, remove it out. I don't know will the expertise allow or not, but would there be any tips or word of caution for those who will try this technique of removing the third floor nuclei? Sorry, not polar body, the pronuclei. Because there would be embryologists who will try to remove the third pronuclei to rescue it after listening to the talk. So, any tips for those embryologists?
So, I'm not sure to understand what you say. Can you repeat me the question, please?
So, my question was, after listening to your talk today, there would be embryologists who would try to remove the third pronuclei to rescue the oocyte. Now, we don't know the skill levels required or the tips required or precautions. So any word of precaution from your side for those embryologists who will try this technique?
Look, at the beginning of the study, we use demicolcine, that is a drug that helps to relax the cytoskeleton of the pronucleus to do it. But after some years, we removed that drug. It's easy to remove. So with really few skills, if you can put, if you are able to do an ICSI, I'm sure that you are pretty good to do epronucleation. At the beginning, we used the demicolcine to remove, to relax, but after that, and use a special pipettes to do that. But now we use the same pipette. Now, now we don't do that. But now you are ready to do this without any drug. If you try to do for skilling, just for trying or for trying, you can put your embryo and try to aspirate the pronucleus with the ICSI pipette inside. Go inside the nucleus, aspirate, and drag. In order to avoid an extra aspiration, so you drag the pronucleus to the area of the syngamy, and when you are out, aspirate the pronucleus and some of the cytoplasms circumvent to remove all the centriole also. So I encourage you to do it. I think you can do it according to your legal framework, and because it's not allowed in all the countries. So you can. I invite you to try, because it's not difficult. Whatever of the audience, I'm sure that is able to do. Let's try.
It's like, it's easy to do. If you are able to perform ICSI, you can do the pronuclei extraction. And there was a question in the box, any specific needle? You have answered that there is no specific needle. It's the ICSI pipette itself which will help to remove the pronuclei. So that question is already answered. Another question which is in the chat is, what is the impact of amount of ulema which gets extracted along with pronuclei? Are there any studies of such impact if you remove out or extract out the ulema, additional ulema with the pronuclei?
To remove the pronucleus and what? To put this pronucleus in another place, you said?
No.
I'm not sure.
The question is, if you're trying to remove the nucleus, there would be some additional amount of oocyte cytoplasm, also known as ulema at times. which will come out in the ICSI needle, right? It may damage the oocyte. Is there any study of impact of removing out additional ulemma along with the pronuclei when you are trying to enucleate?
Okay. If you remove, so I'm not sure to understand what is the question.
Okay. Maybe see once we are trying to remove the pronucleus. So obviously we are going to aspirate some volume of ulemma along with the pronucleus.
If so you aspirate more than you want, that's the point.
Exactly.
Okay.
Exactly.
So that's the fact is that you use the pipette. The important point is you make some negative pressure when you are in the pronucleus, and you try to remove the pipette at the same time. So you are inside the pronucleus, and you try to drag the pronucleus out of the zona where there is the other two pronuclei. So you have three. I can put the video if you want. So there are three pronucleus at syngamy, right? So you put the pipette, aspirate, and when you are sure that are inside the pronucleus that you have decided to remove, you drag, and you are inside the cytoplasm as well. Yet, aún. You are already inside the cytoplasm, but you must to drag to the surface of outside the area when the other two pronuclei are at syngamy. No, I explain myself right or not? And the final aspiration is even when you are with the pipette inside the cytoplasm. So you must to drag, but inside the cytoplasm, you aspirate all the content. You can try, if you try to remove the pronucleus out of the cytoplasm entirely, keeping the structure of the nucleus, the most probably is that the zygote die, right? Because it's a big structure, and there is there is not a large pipette that includes the pronucleus. It must to be so large to penetrate inside the cytoplasm without broken the membrane. So this is why you can go inside with a ICSI pipette and you aspirate all the content, not to remove the pronucleus as a whole structure. Do you understand? I explain myself.
Yes.
The other point is, if you want to remove the pronucleus, you have to do a structure called karyoplast. That means that you have to remove the nucleus with some amount of cytoplasm and surrounded by a membrane. So this is by like a suction, like it's similar. You have to perform a structure similar like a polar body to remove.
Yes. Yeah. That's what I was about to say. It seems quite a bit similar to doing biopsy of a polar body, or even for that reason a blastocyst biopsy. So you pull it
exactly.
You go back and receive back.
Exactly. That's right. So the question is that if you want to remove the pronucleus as a whole structure surrounded by a cytoplasm as a membrane, you need much more skills. And drugs, of course, drugs for relaxing the cytoskeleton. But if the only purpose is to remove the pronucleus without any other else ending for the pronucleus. So I don't want what happened with the pronucleus. Go inside the pronucleus and aspirate it and throw it away. So you don't need the pronucleus for anything. You are not going to reuse this pronucleus.
And how fragile this pronucleus is. Like, is the by any way there any chance that it gets disintegrated and everything, gets disintegrated while pulling it out. So how robust is that pronucleus?
No, I was amazing. I was surprised because at the beginning we used the meclizine, that is a relaxin. I read in the chat. Um, I was surprised when we use this relaxing that the chance of survival, as people ask, was 71% of survival after removal with the drugs. But after removal without drugs, the percentage of survival was identical. Identical. It's similar like ICSI, it's 61%. It's 90% of survival undivided, and finally 40% of blastocysts. So the results were really amazing and they were really good. So the results for survival were identical for me. That's the point, amazing point, is that they were successfully as well with or without the after adding these drugs for cytoskeleton relaxing. So you can do it. You can do it at home as well as you can do it at your lab. You can try it. I would like to if you can I don't want to be an instigator to of illegal things, but you can check your law and your law framework, and if you can, so why not?
Right.
This is a nice point of view for rescue, but not for ICSI, because they become self-corrected by themselves.
Okay, so for ICSI, there is this mechanism, self-correction mechanism that occurs. But we have always been in a practice. It's quite controversial to what we have been practicing actually in our routine practice. So we have always been in a practice of discarding whatsoever. The thing is that if you see 3 p.m. stage, then just discard those embryos.
I think that no one goes to discard anymore, especially because of the new techniques for genetic analysis include also the heteroparental inheritance. So at this point, in fact, I'm not confident at all with the uniformity of the tripronucleated that came from ICSI. This is a point that I have some doubts about how safety it is. I'm not sure that they are all the embryo, all the cells that form the trophectoderm, and all the cells that form the ICM. I'm not absolutely confident with the uniformity of the embryos. I suspect that they could be chimeras, but I'm not sure. So let's wait for them. But now I have a project ongoing, and I'm suspecting that I'm on the right way. I'm not sure about the uniformity. So they try, but I'm thinking about other ways to rescue, but in a more advanced stages, when the cells are discarded. So there is some point that you are haploid, I don't want to be with you. You are triploid, I don't want to be with you. And it seems that a diploid embryo recognized according to the kinetics of the cleavages of the blastomeres, you are not my sister, you are a half sister, you are diploid. And finally the embryo is compiled with all uniform, but we need to study more about this, especially the triple-nucleated.
Right.
And this more space. Sorry.
Yeah, I was just wondering that with time lapse, it has become so easy to observe these things which initially people didn't used to, and this time lapse giving us so much of insights that, as you mentioned, that the ploidy is identifiable by morphokinetics. I believe,
Absolutely.
I don't know, is the day not very far where the AI will also give us the prediction that is this haploid, diploid, or triploid.
Absolutely. The time-lapse technology changed our lives. Our lives as embryologists. We thought that we know a lot, but we don't know nothing, anything. So we're learning every day new things, and when you is to be live there, you can be here like this at the we were time and time watching movies and movies, the embryos, and every time you are learning, the time lapse is always teaching us more and more about embryology. So I think that to have 1 or 2 time lapse at your lab is more than recommended, because you're always learning, and it's always your mind is on fire all the time. She is feeding your knowledge all days, and it's very useful for these things, as you say, for predicting those diploid, those triploid. I think that it is because morphologically they are equal, absolutely equal, but the timing is not.
Right. Yeah, I mean, morphokinetics will literally help us a lot. And obviously, with the future machines coming with morphokinetics being also studied along with metabolomics, I think the synchrony will help us to explore more of the embryology, because earlier days, there were a lot of scientists who used to believe that the fourth check is essential and you should use only 2pn. Then came on a lot of papers because of time lapse that the self-correction mechanism is there. You have 1pn and 3pn, which also ends up into euploidy. And then it was realized, okay, 2pn is not everything, though the chances are very less that you may have 1pn, 3pn giving a good pregnancy, but it is a chance. And the patients who are actually coming to you for infertility treatment, that even 1% of the chance cannot be missed if you can deliver a baby out of it.
Right. Exactly.
So that
it's very precious, especially for the cases where we have very limited number of oocytes. So it is very precious.
Yeah, and even now, you with the risk that it's supposed that now this going to starting is starting this AFO for clinics. So, but when especially with triple nucleated, or when the patient is going to plan the reproductive project, patients, the good practice is to cryopreserve, and if the chance to obtain a monopronuclear improve because of I vitrify my oocytes, and why I have to discard one possible embryo that is good for me. So I I try to rescue everything, immature, mature, monopronuclear, everything.
Right. That's a perfect combination of a scientist and an embryologist trying to get the best out of everything.
And I want to add one thing to your comment, that was that, of course, the proteomics, but also to make the cell tracking and to know how this cell, when start here and finally this cell goes to the embryo here, or goes to the blastocyst at that point, or is not included or is excluded at the morula compactation or even at the blastulation, how this behavior of the cells to know. I sometimes I wish to have a rubber or to have a color and to paint this cell, and know I want to know where are you going to be in the next 2 days to follow your progeny, how are you going to go. This is a good point that the technology has to improve too, at the point of the time lapse. Almost for me, it's important. Of course, the proteomics and whatever non-invasive technique that you add, the best for the patient, the best for our knowledge, the best for the results.
Right. So I think one of the last questions, which also is there in the chat box, is what is the percentage of chances of forming a blast after removal of after enucleation of one of the PN from a 3PN zygote?
Our percentage was, if you remember the publication that was published in the Fertility and Sterility in 26, it was the chance of blastocysts. It's when you perform the e-pronucleation, 71% survive, and after culturing them, 40% reach the blastocyst stage. That percentage was similar to that achieved by currently fertilized oocytes. And it's twice that the blastocyst rate achieved by those non-manipulated triple-nucleated IVF zygotes. So triple-nucleated, not microsurgically, non-microsurgically epronucleated. So intact triple-nucleated from IVF has a 20% of blastocyst rate. When you remove the surplus male pronucleus, the chance increased up to 40%. It's similar to that observed in currently fertilized oocyte, that is around 50-54%.
Yeah, that's quite similar.
And the most amazing aspect is the morphology, because the morphology is amazing. It's totally different. It's totally different.
And not only morphology, and as you said earlier, that all these embryos should be subjected to PGT examination as well.
I suggest always. I'm pro PGTA. I'm pro. I'm so sorry, but the chance and the time is and that's the money is an economic question, and I think it's the opportunity for the patient is no negative betas, is to achieve the success in a less time required, in a less money, in a less number of transfers. So I'm pro-analyze. I support the analysis of the embryos, of course. For me, is in my opinion, because I as all the audience agree with me, is that as you agree, how many lovely blastocysts became into abortion? How many lovely, pretty trophectoderm, lovely blastocysts that you say, oh, this is the one, and you put it in a lovely also transfer, and this is a 0, or this is an abortion. So this is a failure for us and for the patients. And this time and one negative and another, an abortion is a long effort for all. So I prefer to know what I put in. I know to know this is good, and I know that this is good. I have an information that said this is the best one, so this is more confident for me.
Yes, nobody is perfect. So even a blastocyst with a beautiful grade cannot be perfect. Sure. Yeah. And your discussion also already gave answer to the next question, which was in chat box, that it's already been answered, that yes, if it is 3PN, you culture them, and if it becomes a blast, you send it to the genetic testing, and accordingly you act. But that also has one question, I mean, which came into my mind. You said you are pro PGTA. You support PGTA, but I am also aware that there are a lot of papers which you have also done with Dr. Carmen Rubio, who is into non-invasive PGTA, and with 3PN correction. What are the chances that non-invasive PGTA may also detect what the PGTA is detecting on the front of 3PN, 1PN?
I have a revision published about this topic, and I'm not pro PGTA in the media. I'm pro PGT invasively. I'm so invasively for PGT analysis. If you want to give more info about the, so if you provide proteomics, if you provide hyperspectral imagings, if you provide whatever tool that helps embryologists to select the best one, is welcome, of course. Of course, it's welcome. The non-invasive, by proteomics, by whatever technology, for sure. But never as a substitute, never. For me. So the time-lapse technology, the IDA score, the whatever tool is more than welcome. Of course, yes, of course, but never for substituting the one technique that is, see, if the people is controversial about the Mosesism, about the chimerism, about many things, and why to what is the how is the trifecta uniform, or how not is doesn't like. So invasive, I prefer to be invasive, and for not invasive as a tool for help embryologists, sure, but not for substitute the genetic DNA analysis that came from the cell that they removed from the blastocyst. That's my opinion. We have a pretty review about this, a non-invasive PGTA that has published it maybe, I think, 4 years ago. And the results go slower than expected. So.
Okay. Great. So, I think thanks for such an excellent session. It was thorough science.
Yes, I can see so many comments
on. You are more than welcome.
Yes. It would be great to listen to you in the oral session, which you have put across that it has been selected in ASHRAE. We would be there. Obviously, I would try to come down and listen. It would be, I suppose, an excellent and exciting talk. We'll definitely try to meet you over there.
Okay, when the audience, if some of the audience want to or has engaged to this challenge of epinucleation, the next talk came and share with us the experience. If your group goes to the epinucleation, as it's remove the mind of the people, I'm a pleasure to share that person share with us the results and the experience with the epinucleation of triple nucleated embryos.
Great. Thank you so much. Thank you so much for bringing that.
Yes, Maria, for taking out time from your routine and enlightening our, giving us such an enlightening session to our audience. And we really appreciate this thing.
And the good part was that the message was not only about how to rescue, but we could see how she's working, like rescue, do everything best for the patient, whatever is coming to embryologist, let it be PhD, NIPhD, time lapse, or any other thing. But an embryologist should think always for the betterment of the cycle outcome for the patients and work accordingly. That passion which you were, the passion could be seen while you were delivering the talk and answering to those questions.
As I said, is all the technological background put on the focus, the patient. All the technological background that you have, all of them.
Right. Great. Thank you so much. Pleasure to have you. Thank you again, IFA. Thank you, Sir.
Thank you very much. And it was an initiative. Again, I will just like to repeat this thing. It was an initiative by IFA. So IFA holds this international webinars every third Friday. So we'll be meeting next Friday, third Friday of July, with some new speaker, with some exciting new topic. Until then, goodbye, and wish you a happy ASHRAE. Wish you a happy ASHRAE. Let's have a meetup at ASHRAE. Those who are coming will be, definitely love to attend your seminar there as well.
Thank you so much, and have a good weekend, all of you.
Thank you.
Thank you.
Bye-bye.
Goodbye, everyone. See you next third
See
Friday.
you next Friday. We talk to you next Friday.
Yeah.
Okay. Next Friday, 1st of July. Is day 5?
It's third Friday.
One Friday, Friday in July.
What is the date coming on third Friday?
Which day? I don't hear right. July.
Sir, I think 15th or 16th of July.
15th July.
July. So third
The
Friday is
19th July.
19th July. So 19th July we will be meeting.
19th
19th July.
July. Okay.
Yeah, yeah.
Pleasure. It's a pleasure for me.
Thank you.
Okay.
Thank you very much.
Thank you.
Have a good weekend. Bye bye. Thank you so much.
Thank you. Thanks, man.